All right, hello 12s. Continuing on our discussion of eukaryotic cells. Let's talk about now. So we've been talking about membrane-bound organelles, right? So those have the plasma membrane. They're the sacs of lipid, basically, and stuff is happening inside those sacs of lipid. So that's the nucleus, the nuclear membrane, the endomembranous system, so endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum, the Golgi body, vesicles. Then we had those chloroplasts in plant cells. Mitochondria, also in plant cells and animal cells. Mitochondria, the powerhouse of the cell, doing the Krebs cycle, producing 36 ATP in those mitochondria. The chloroplasts, using the light reactions to make oxygen and ATP so that it can take carbon dioxide and turn it into sugars, into starch, or parts of the plant, or used as energy by the plant. And those were all membrane bound. So they all have the plasma membrane. They all have the double lipid bilayer with the polar hydrophilic phosphate head and the fatty acid tails facing each other. And the inside here is where my nose is non-polar. So water can't get in. But there are other organelles that are non-membranous organelles or non-membranous cell structures. So these don't have a cell membrane. And these you will find in eukaryotic cells, so animal and plant cells. Not all of them, but some of them, one of them specifically will be in all cells. And a lot of these are also found in bacteria cells, in prokaryotic cells. So let's talk about some non-membranous. So the first one, that is the most important one, and you will find in every single cell type on Earth, is the ribosome. Ribosomes. Okay, and what ribosomes are is why they're so important is they're the site of protein synthesis. And protein synthesis is making proteins from DNA. So taking DNA, turning it into mRNA, and then that mRNA travels out of the nucleus and combines with a ribosome, and the ribosome is where that protein gets assembled, where it gets read. Ribosomes are made actually from RNA. They're made from ribosomal RNA and protein. Ribosomal RNA and protein combined together to make the ribosome. And they are made in the nucleus. The ribosomes are made in the nucleus. The parts of them are made in the nucleus. It's sort of like, certain cars, like German cars. The parts of them are made in Germany, and then they actually go to another country to get assembled. I don't know if that makes sense. I think Japanese cars, they do that. In Ontario, they assemble Hondas in Ontario, but a lot of the parts are made in Japan, and then they're shipped over here and they're assembled. So the same thing. That doesn't make any sense. That doesn't help you, but whatever. So the nucleus is where the parts of the ribosome are made, and then they travel out of the nuclear pore into the cytoplasm or into the rough endoplasmic reticulum, and they're assembled. So remember, the rough endoplasmic reticulum is rough because it's full of ribosomes. Those little bitty bits, they're full of ribosomes. So let's see here. Can we? Okay, well, you'll have to look. I don't know if you guys can see it. Something's weird. Oh, it's in blur mode. Okay, whatever. So there you go. There's the subunits of the ribosome. There's two. There's a large subunit and a small subunit. And these would be made inside the nucleus, and then they travel through a nuclear pore out into the cytoplasm or to the rough ER. Remember the rough endoplasmic reticulum? Oh, there we go. There's mitochondria. There's some rough endoplasmic reticulum right there. you can see all those little dots. Those are ribosomes. So those are going to be where proteins are made. And you can see there's millions of them because your cells need lots and lots of proteins. So there's going to be lots of them, all the enzymes, all the proteins. So these are really important. That's why all cells on Earth will have ribosomes, because all cells need to assemble proteins. Otherwise, they wouldn't exist. They wouldn't work. We're proteins after all. So yes, ribosomes made of ribosomal RNA and proteins. And the subunits are made in the nucleus. They're made in a specific part of the nucleus. Let's scroll here to the specific part of the nucleus. There we go. Is the nucleolus. Here, I don't know, focus on that. There we go, the nucleolus. So there's the whole nucleus. Oops. There's the entire nucleus, and there's the nucleolus. It's the dark spot in the nucleus. In prokaryotic cells, it's called, they have a little dark spot too. Prokaryotic cells, remember, don't have a nucleus, but they do have a little dark spot of DNA and stuff that produces their ribosomes, and it's called the nucleoli. There is a structure called a polysome that's in the cytoplasm. So most of these ribosomes you're going to find in eukaryotic cells in the rough endoplasmic reticulum. That's where those peptide chains get assembled. But there's also these things called polysomes. And what polysomes are, they're structure of ribosomes where if you need to make a whole ton of protein all at once, what will happen is, do I have a picture? I don't have a picture of it. I don't think I have a picture of it. I thought I did. It would have been good to have a picture. Basically, imagine beads on a string. And the string is mRNA. So you'll have a long chain of mRNA. That'll be one gene. That'll be the code for the protein. And then what you'll have is you'll have ribosomes right along that mRNA, a whole bunch of them, reading one mRNA strand, assembling a whole ton of protein. So one ribosome will be making that protein. Then a second one will be making that protein. A third one will be making that protein, all from one piece of mRNA. And these proteins, so they're in the cytoplasm. They're not going to go into, these are going to be proteins that are used in the cytoplasm. I would say a lot of these proteins are going to be proteins that are part of like the cytoskeleton. Often the cytoskeleton has to be built really fast. So they might not have tertiary structure, because if you think about it, they're not going through the ER, the endoplasmic reticulum, and then some tertiary structures made in the endoplasmic reticulum, and then a lot of it is made in the Golgi body. That's where cells are folded the rest of the way. Here, they're in the cytoplasm. These polysomes are in the cytoplasm, this string of ribosomes that is just reading it. So some of that processing might not happen for those proteins. So these would be cytoskeleton. Let's talk about the cytoskeleton. That's a good transition for the cytoskeleton. So that's another non-membranous structure. So ribosomes don't have a cell membrane. They're just made of protein and ribosomal RNA. Cytoskeleton is made from protein that has only that secondary level of structure. It doesn't have that tertiary level. I don't want to say that for sure, because nothing in biology is for sure, but a lot of cytoskeleton doesn't have that tertiary structure. And then what these are, so you got microtubules, microfilaments, and microgranules. We're not going to go into too much detail on this. We just don't have time to go into horrible detail on this, but they make up together all of these things. They combine together to make up the cytoskeleton, and they're different sort of versions of proteins. And so the cytoskeleton is what anchors the insides of the cell. It's these scaffolding. So like the mitochondria and the nucleus and all those organelles, they don't just float in there. Like it's not just a sack of floating lipids. They're actually anchored in places by the cytoskeleton matrix that spreads out throughout the cell. Let me see if I can ask cytoskeleton in a eukaryotic cell. Just asking Google to show me a picture of cytoskeleton so you guys can see. Here we go. Here's something. This will kind of do. So here you can see There's the nucleus, and all these little things, that's all microtubules and cytoskeleton inside. It's like a scaffolding, a matrix of it. And there might be, let's see if we've got any like pictures sometimes, because what we can do is we can actually attach antibodies, which I don't know when we're going to learn about that. That glow. And we can look at the cytoskeleton. Here we go. That's what I want. So there you go. Oh, yeah, that's perfect. So there you go. You can see that's an actual cell. And you can see all that stuff. That's antibody. That's glowing cytoskeleton. So you can see it makes quite a thick matrix. And the cytoskeleton will do a whole ton of stuff. It will, it can be super important in cell division. Right, is you've got to take apart the nucleus, so pull things apart, separate the organelles out. So half the organelles go to one side, half the organelles go to one side. The DNA gets lined up at the center. The chromosomes get lined up at the center, and then they get moved to the sides. That's all coordinated by the cytoskeleton, and those polysomes are going to be important in manufacturing new cytoskeleton. Those little they're making these little tubules that then combine together and move along. Yeah, that's you got that. You'll understand that. There's some other parts. So cytoskeleton is made up of microtubules, microfilaments, microgranules, all these little subunits, proteins. Also cilia and flagella. So certain cells, our cells have cilia and flagella. And you'll also find these in prokaryotic cells, cilia and flagella. So flagella are the big whippy tails. So like sperm cells have a big whippy tail. That's a giant flagellum that they use to propel themselves through, to move. And cilia are lots of little hairs, lots of little tiny ones. So they do this. And they can move a cell or move fluid through organs and stuff. So they can be used to move, or they can be to move the cell, or to move the fluid around the cell. So some eukaryotic, single-cell eukaryotic organisms have near their mouth, they'll have cilia, and they suck the fluid in that way. They move the fluid. We have cilia all down your trachea, all down your throat. You've got cilia that move mucus up your trachea, which leads to your lungs. They move this mucus up. They're constantly beating to move mucus up into your pharynx, into the back of your throat, so that then you swallow that mucus. And the reason for that is because your lungs are vulnerable to disease. They're vulnerable to pathogens. So these pathogens, they fall in, they get stuck in the mucus. The cilia move that mucus up. You swallow it, your stomach acid dissolves all those pathogens. That's why smoking is so bad. Vaping, smoking, whatever, because what it does is it hurts those cilia. It kills, dries them out, and kills them. Smoking covers them in tar. Even smoking pot cigarettes does the same thing. It burns them, it hurts them. And so then what ends up happening is those pathogens can make it down into your lungs, infect your lungs, and you always have people who smoke a lot, they're always a little like, that's because they've always got a chronic lung infection kind of going. So don't hurt your cilia. Now these flagellum and these cilia, they're anchored into they have to be anchored into the cell's membrane, right? Because they're this huge thing. And so the anchor that they have, it's called a basal body. And they actually grow, cilia and flagella grow out of this basal body. And it's just a collection of microtubules that is embedded in the cell membrane and attached to the cytoskeleton, so that flagella or cilia can grow out of it. It just anchors flagella and cilia. There's something also called a centrosome. So basal bodies, that's going to be an organelle, that's a non-membranous organelle that flagella and cilia grow out of. There's also a centrosome, and the centrosome Here in the image, there we go. It's centrosome. It's very hard to get this. There we go. Centrosome is there. This is all in the notes. There it is. It's near the nucleus, and that coordinates the cytoskeleton. So the cytoskeleton grows out of the centrosome, can grow out of the centrosome. It also can grow the basal bodies. It's sort of like the precursor, the starting of microtubules and these bits, microfilaments. I don't go into too much detail of this. There's microtubules. The last non-membranous organelle that we're going to talk about very little, is a very short bit in the notes here, is cell walls. And cell walls, you're not going to find them in animal cells. You're only going to find them in plant cells. So plant cells have cell walls. And these are basically sort of protein and sugar. They're dead. They're not alive. They're like the plant cell secretes it out. What can I say about it? It gives structure and protection to plant cells. Bacteria cells, prokaryotic cells also have cell walls. They're made out of something different though. They're made out of peptidoglycan, but plant cells are made of like cellulose. There's advantages and disadvantages. The advantage is they protect the cell, prevent it from drying out, give structure to the cell so you can grow giant trees or you can sit on cell walls. But the problem is they limit the cell's ability to move things in and out of the cell. It's hard to get things in and out because they can't cross the cell wall. There's little holes in the cell walls that they can move in, but so it limits the surface area basically of things getting in and out. As a result, metabolism has to slow down. Plants don't have a fast metabolism. They can't react very quickly. We can because our cells are constantly moving stuff in and out. Plants, no. They can't get too excited because then they would require too much sugar, too much speed, and that stuff couldn't get into the cell fast enough. And the waste couldn't get out fast enough. So metabolism in plants is really slow. Plants grow slowly. They don't move very fast. All that. But they have cell walls to protect the cells so they don't have to move very fast because it's hard to hurt them. That's their strategy. So that's the disadvantage, is that things can't get in and out very fast. And as a result, plants have to store a lot of their food and they have to store water inside the cell. So humans, we have vacuoles as well. So these are basically they're membrane-bound sacs that store water, but they're very small. In animal cells, they're small. They're just like temporary. They're basically lysosomes. Little vesicles, let's call them vesicles. Whereas plant cells have huge vacuoles. Sometimes almost the whole cell in the plant you may have seen will have a huge vacuole that stores food and sugars, and wastes can be dumped in there too, waiting to get out of the cell if there's some you can't get out fast enough through the plant, so it's not hurting the cytoplasm. That's it. That concludes eukaryotic cells. So we've talked about the nucleus. We've talked about other membranous. We talked the endomembranous system, ER, smooth ER, rough ER, Golgi bodies, lysosomes, endosomes. We've talked about mitochondria, we've talked about chloroplasts, now we've talked about ribosomes, we've talked about cytoskeleton and microfilaments, microtubules, the little bits of protein that make up the cytoskeleton, and cilia and flagella, and finally cell walls. So there you go.